What is the latest in epilepsy genetics? Based on a review article by Rougero et al. 2023, the majority of epilepsy is known to be attributable to genetic factors, as has been long established. Genetic variation is thought to explain up to two-thirds of the overall liability to epilepsy in the general population, and genetic susceptibility can be attributed to monogenic, polygenic, and other multifactorial contributions. Vast progress has been made in the past 20 years in the identification of the genes causative of epilepsy, as well as the characterization of the phenotypic architecture of many of the genetic epilepsies. The first gene to be identified as causative of epilepsy, ChRNA4, was identified in 1995, and now, over 15 years later, over 1,000 genes have been identified that have been associated with monogenic epilepsy. In addition, it has now been established as standard of care to complete a genetic workup for individuals with unexplained epilepsies, even individuals who have typical development in epilepsy that is well controlled. Whole exome sequencing is now recommended as a first-line test in individuals with developmental delay and intellectual disability, conditions where genetic contributions are known to be highly comorbid with that of epilepsy. With this huge advancement in possible genes to identify, as well as people in whom these findings can be identified routinely, the field of epilepsy genetics has experienced an explosive growth in available knowledge and new questions to answer. Progress in identification of genetic bases of the epilepsies. 10 years ago, the field was revolutionized by novel high-throughput methods that led to the discovery of what is still the most common known cause of monogenic epilepsy, PRRT2, which causes self-limited familial infantile onset seizures, as well as the discoveries of the monogenic causes of malignant migrating partial seizures of infancy, LOSMPSI, and autosomal dominant nocturnal frontal lobe epilepsy, ADNFLE, KCNT1. Genetic etiologies are still being discovered now as they were then through large family studies and international gene matching collaborations. Though in addition, international data-sharing collaborations and large exome sequencing studies performed on thousands of individuals, such as EPI4K and EPI25 Collaborative, the Deciphering Developmental Disorders, DDD project, and cohorts analyzed from clinical testing labs, are creating massive data sets that have the power to allow new gene discoveries to rise to the surface. While gene identification is essential, it is just as imperative that evidence supporting gene-disease relationship emerge from both genetic and experimental evidence. Over time, evidence has accumulated significantly to create definitive or strong disease-gene relationships for genes causative of epilepsy, as has been systematically analyzed by Clinical Genomics Resource Epilepsy Expert Panel. Novel gene discovery. Hundreds of new epilepsy genes have been discovered in the past decade that explain epilepsies that cross the spectrum of self-limited epilepsy to developmental and epileptic encephalopathy. Diagnoses for children with DEEs now can be identified over 40% of the time due to the steady advancement of gene discovery and characterization. Collaborative international networks have led to the discovery of further relatively common genes causative of DEs, such as SCN3A, which is causative of a severe childhood onset epilepsy, sometimes with malformations of cortical development. In addition to SCN3A, further ion channel genes have been identified as causative genes for human epilepsies in the last 3 years, gradually unveiling the full landscape of epilepsy-related channelopathies. KCNC2, encoding a member of the Shaw-related voltage-gated potassium channel, or KV3, family important for high-frequency repetitive firing of action potentials, was identified as a rare cause of developmental and epileptic encephalopathies and generalized epilepsies. Both gain and loss of function variants were identified as disease-causing, and in contrast to phenotypes associated with the closely related KCNC1 gene, none of the reported individuals have been found to have progressive myoclonus epilepsy. In addition, biallelic loss of function variants in CACNA2D1, encoding the auxiliary A2D subunit of voltage-gated calcium channels, were identified as a rare cause of severe developmental and epileptic encephalopathies. The synapse disorders represent a second gene group with constantly increasing number of disease genes implicated in human epilepsy. In addition to the discovery of AP2M1, encoding the M subunit of the adapter protein complex 2 involved in clathrin-mediated endocytosis and the synaptic vesicle cycle, SNAP25 and UNC13B, and have been identified as genetic causes of human seizure disorders in the last 3 years. In contrast to UNC13B, which was found as a cause of a mild focal epilepsy, both AP2M1 and SNAP25 cause developmental and epileptic encephalopathies or complex neurodevelopmental disorders. Other notable genes identified in the last 3 years include SPTBN1, encoding a neuronal BII spectrin given its relatedness to SPTAN1, one of the earliest identified genes for developmental and epileptic encephalopathies, listed as developmental and epileptic encephalopathy 5 in OMIM. In parallel to SEPTAN1, the phenotypic spectrum of SPTBN1 disease-causing variants includes severe epilepsies and neurodevelopmental differences. An additional gene worth noting includes the SLC32A1 gene, encoding the vesicular inhibitory amino acid cotransporter VGAT. The SLC32A1 gene is notable as it is one of the few recently identified genes for familial epilepsies inherited in an autosomal dominant manner. Specifically, disease-causing variants were first identified in families with genetic epilepsy with febrile seizures plus, GEFS plus, followed by the identification of a broader phenotypic range due to de novo variants that includes developmental and epileptic encephalopathy with a prominent hyperkinetic movement disorder. The genetic architecture of the epilepsies is built through up to 20 genes recurring commonly across many people and families, and then a long tail of remaining genes, each explaining no more than a fraction of a percent of genetic epilepsy on their own. Most genetic discoveries of recent identify individually rare, but collectively common conditions. There are the genes that can cause a spectrum of epilepsies based on differing genetic variants and factors yet to be discovered, such as KCNC2, which can lead to both genetic generalized epilepsy presentations and depresentations. NPRL3, a modulator of the Gator complex, an mTOR regulator within the brain, has been identified as causative of a spectrum of focal epilepsies, which can run in families with variable penetrance and expressivity. This gene has been found to explain a much higher proportion of familial and non-familial focal epilepsy than could have been predicted with the gene's discovery in 2016. Perhaps this phenomenon could be explained by the increased testing available for individuals who have self-limited epilepsies, and underscores the importance of testing availability for individuals with self-limited and focal epilepsies. Finally, new genes have been discovered through novel methods, such as phenotypic similarity analyses, while genes are consistently discovered through genotype-first approaches, such as massive sequencing efforts that allow for identification of rare and de novo variants in novel genes. It has been demonstrated that phenotype-first approaches can be used to identify new epilepsy genes. AP2M1, a gene causative of neurodevelopmental disorders with generalized epilepsy, especially myoclonic-atonic seizures, was discovered through analyzing phenotypic similarities constructed through phenotyping over 300 individuals with DEEs with the Human Phenotype Ontology, HPO. These novel approaches to gene identification will continue to grow as the phenotypic variability of the epilepsies is further defined. Polygenic and other genetic mechanisms. Novel disease mechanisms, including pathogenic intronic variants and repeat expansions, and innovative integration of genetic data with polygenic risk scores, have created paradigm shifts in the field. Non-coding variants within an intronic region of SCN1A were found to promote inclusion of a poison exon, causing Dravet syndrome through reduced SCN1A expression. Intronic expansions in SMD12 and other genes were identified as the cause of adult familial myoclonic epilepsy, FAME, a borderland condition between myoclonic epilepsy and tremor that was found to affect hundreds of individuals given the familial inheritance in this condition. Since a large percentage of the genetic liability for generalized epilepsy is expected to be explained by common genetic variants, polygenic risk scores may reflect a significant contribution to the overall population risk. Despite providing valuable information about risk, Polygenic risk scores do not indicate underlying biological causes of disease and do not appear likely to ever do so, because the risk signals are spread broadly even through the genome. For this reason, it appears unlikely that therapies can often be effectively targeted on the basis of individual polygenic risk scores. Polygenic risk assessment is also currently limited to specific populations. Recent progress has been made in understanding polygenic epilepsies through genome-wide association, GWAS, and polygenic risk analyses. Recently, new genetic factors related to propensity towards febrile seizures have been identified, collectively explaining 2.8% of the variance in the liability to febrile seizures. Genetic factors involved in this propensity include those that regulate response to fever, as well as genes that influence neuronal excitability through ion channel, SCN1A, SCN2A, ANO3, and synaptic transmission, BSN, ERC2, GABRGS2, HERC1. Over 7,000 cases and 83,000 controls were analyzed to attain these results, reflecting the massive numbers of research participants that will be required to further define and investigate polygenic risk. Complex inheritance of multiple common variants in key genes also have been analyzed using data from international consortia for sporadically affected individuals, as well as people with familial epilepsy and their unaffected relatives. This analysis was able to demonstrate that individuals with familial epilepsies, or 1.20, and sporadic epilepsies, or 1.09, had an elevated polygenic risk compared to controls. PRS scores were most pronounced in familial cases of GGE, or 1.58, emphasizing the importance of familial aggregation, particularly in the generalized epilepsies. Novel presentations of known genes. It has been repeatedly seen within the field of epilepsy genetics that monogenic explanations are found first for severe developmental and epileptic encephalopathies, and then mild and novel presentations are later attributed to the same gene. Both STXBP1 and KCNQ2-related developmental and epileptic encephalopathy, DE, were first identified as causes of Ohtahara syndrome in neonates who went on to experience intractable seizures across their lifespan. These conditions now are known to cause much more variable presentations of epilepsy and developmental phenotypes, including infantile spasms without prior neonatal seizures, and sometimes neurodevelopmental disorders without seizures. As more individuals are analyzed through genetic workups, explanations for more mildly affected individuals with epilepsy are increasingly being identified. Mild variants in genes once thought to be causative of DEEs in all cases are being found in individuals with self-limited epilepsies and more mild impacts to their learning and development, as has now been reported rarely in STXBP1. Further, novel disease-causing mechanisms and variants within known epilepsy genes are being identified. DNM1-related disorder, a severe DEE characterized by profound hypotonia, highly abnormal hypsarrhythmic EEGs, West syndrome evolving to Lennox-Gastaut syndrome, and severe to profound developmental delays, was first identified to be caused by missense gain-of-function variants within the ATOP, AS, and middle domains of the protein, interfering with the protein's ability to heterodimerize and aid in vesicle recycling. Recently, the most recurrent described variant in DNM1 was identified, an apparent splice variant, C1197-8GA, in the canonical isoform of the protein, but one that actually acts as a non-apparent missense variant in the isoform of the protein most highly expressed in the pediatric brain. Additionally, genetic etiologies first characterized as operating through dominant negative or loss-of-function mechanisms have had novel variants identified that are proven to operate in a gain-of-function manner, resulting in novel presentations and phenotypes. Novel variants in SCN1A have recently been shown to cause a severe early-onset phenotype diverging from Dravet syndrome, characterized by epilepsy that onsets significantly prior to the classic 5-6 months of age, and which results in a unique developmental profile. Progress in variant analysis and interpretation. Novel approaches to variant databasing, aggregation, functional testing, and interpretation are growing exponentially in recent years. which is a critical step towards making data available and interpretable to those providing clinical care. Variant and functional data has been grouped into publicly available portals for the NMDA receptor family of genes, SLC6A1, and the sodium channel family of genes. These browsable resources allow for the combination of genetic, phenotypic, and functional data for improved variant interpretation and VUS resolution. Functional characterization of a variant's effect on a protein is essential to establish the underlying pathobiology of the genetic condition to create opportunities for precision therapy, as well as to resolve variants of uncertain significance. Large collaborative efforts within the NIH-funded Centers Without Walls, CWOW, have performed mass characterization of variants within multiple epilepsy genes, as was recently demonstrated in KCNQ2. These studies create the basis of pathobiology for these variants, and at times identify paradoxical effects on channels that have led to the discovery of novel presentations of known epilepsy genes, such as that seen in SCN1A. This increased knowledge of variant impact and functional analyses has led to increasingly mounting evidence proving gene-disease relationships. Progress in understanding the clinical picture of genetic epilepsies. In parallel to the explosion of the understanding of genotypic data that has occurred over the last decade, it has become increasingly imperative to characterize the phenotypic architecture of genetic epilepsies being more routinely diagnosed. Our understanding of the genetic underpinnings of epilepsy has thus far outpaced our understanding of phenotypic architecture and variability. Outlining the natural history and progression of genetic epilepsies is a massive undertaking requiring a multi-pronged approach, including case series and large family studies, natural history studies that follow smaller groups of children affected by a genetic condition in granular and comprehensive detail, and finally the use of computational methods such as phenotypic reconstruction through HPO and electronic medical record (EMR) data to characterize genetic epilepsies based on thousands of years of patient data that already exists, waiting to be analyzed. Natural history studies for Rett syndrome, Angelman, Dravet, and CDKL5-related disorder have shown prime examples of deep phenotypic description and characterization of affected individuals over time. Parallel to deep phenotypic characterization in smaller groups of affected individuals are efforts to aggregate existing data from the literature in addition to data captured in routine clinical care in the EMR, which allow for enough power to bring new features and genotype-phenotype correlations to light, as has been seen in STXBP1, SCN2A, KCNT1, and SCN8A. Finally, reconstruction of genetic diseases through HPO-based methods has allowed the field to place computational power behind static and longitudinal phenotypic description, leading to novel discoveries. This emerging field of phenomics allows for static and longitudinal disease reconstructions that will allow for identification of natural history study and trial endpoints in the future. These joint efforts are leading to a comprehensive understanding of the complex phenotypic architecture of the epilepsies over time. Progress in precision medicine. With diagnoses being identified in an increasing number of people with epilepsy, the question remains as to the extent to which treatment is affected by genetic diagnosis. Through systematic analysis of this question, it has been found that genetic diagnosis can influence treatment in nearly 50% of cases, through the influence on treatment options, including anti-seizure medication, ASM choice, and the facilitation of preventative and specialty care due to known risks associated with particular genetic diagnoses. Through the analysis of over 400 individuals, it was found that new medications were added or initiated in over 1/3 of individuals following genetic diagnosis. Many genetic diagnoses causative of epilepsy have an associated precision medicine therapy that is recommended or suggested post-diagnosis. For example, carbamazepine is recommended as a first-line medication for people with infantile seizures due to PRRT2, but also for adult-onset dyskinesias often present in this disorder. Sodium channel blockers are recommended to be avoided for loss-of-function variants with sodium channel genes such as SCN1A, SCN2A, and SCN8A, but they have been trialed with success in individuals with gain-of-function variants in these genes. Considering diagnostic rate of genetic testing in individuals with epilepsy across all testing modalities has been found to be 17% and much higher in individuals with developmental and epileptic encephalopathy and neurodevelopmental disorders. A significant proportion of the pediatric and adult population with epilepsy has the potential to benefit from a clinical genetics evaluation. Though this potential exists, it should be stated that, for most genes causative of epilepsy, a precision medicine is not available or has been found to be less effective than initially theorized. Future research will seek to compare effectiveness of known and novel epilepsy medications in individuals with genetic diagnoses. In addition, the first gene and genetic therapies are now trialed for distinct genetic epilepsies, including for SCN1A-related Dravet and KCNT1. However, at the writing of this review, it is critical to point out that none of these studies have been published, a caveat that is critical given the high and sometimes unrealistic expectations for novel treatments. Gene therapies are under development for several genetic epilepsies, though it is still uncertain as to how these novel approaches will be delivered safely to the central nervous system and the robustness of an effect they might have. Conclusion. Through international collaboration between clinicians, scientists, and family advocacy, knowledge of the genetic epilepsies continues to grow exponentially. In the clinic, genetic testing for individuals with any unexplained epilepsy has been officially set as standard of care by the National Society of Genetic Counselors, NSGC. Gene discovery continues at a steady pace, and we have a better understanding than ever of the natural history of genetic epilepsies, gained through gold standard and innovative methods. We are seeing the first gene modulatory therapies being trialed in the genetic epilepsies, and vast amounts of research focused on precision medicine approaches. Continued efforts of collaboration, data sharing and harmonization, and integration of the massive amounts of data that has been collected in the research and clinical domains over the past years will underpin our future discoveries and understanding. Key points. 1. Genetic testing for all individuals with an unexplained epilepsy is now standard of care, with exome sequencing often being the most appropriate test of choice. 2. Increased genetic testing of individuals and huge multinational exome sequencing and data sharing efforts have led to further genotypic and phenotypic characterization in the epilepsies. 3. Collaboration with disease advocacy organizations has pushed the field of epilepsy genetics and precision medicine forward. Thanks for listening, and I hope this is helpful for you. Dr. Hamza Al-Sayyouf, child neurologist, American Board, Amman, Jordan.